Okay, hello everyone. First off, we are the iGEM design team, and this is specifically the lab committee portion of the iGEM design team. In these series of videos, I would like to go over some of the fundamentals of what we do in lab in order to catch everyone up to speed to get people prepared for the stuff we're going to be doing in lab and for competition year. In these series of videos, the later recordings are going to be before this fact. So I'm recording this way after those original recordings were recorded. Because of that, I can record these first few videos with a new sense of where this whole thing's going. Just keep that in mind, though, because after episode 3, there's going to be a cut to presentations during the lab meetings themselves. There's going to be an audience. There's going to be people talking. So keep that in mind. I thank you for watching these videos, and I guess we'll begin. So during each of these slide presentations, I have a little index where we'll go through what we're going to cover. First, I'm going to touch the lab canvas training for trainees. So any new lab member has to go through the lab canvas course. It's a series of modules that you take in order to get a general understanding of what we do in lab done. It's pretty basic. It's a series of multiple choice and fill in the blank questions that you take. There's some reading and maybe some video watching as well. Also, during this lab canvas course for trainees, you will become familiar with the lab manual, which is the main document that we use within lab. It outlines all the different protocols that we use in lab, all the knowledges that we need in order to understand those protocols. And also it has some equipment and how to use the equipment. Finally, I'm going to be covering some iGEM fundamentals for this video. Those include what DNA is, what RNA is, what transcription is, and finally what translation is. For those of you that have taken biology already or just some fundamental level science in high school, I assume that this is going to be pretty basic to you. If you want, you can skip through it if you know all this stuff already, but this is just to get everyone up to speed. Okay, so first are the modules. So modules are broken into different subcategories. The first category is the introduction. The introduction tells you the purpose of the module on the Canvas course. Each module is split between what they're covering. So say the first module covers how lab is structured, and the second one covers DNA. So within the introduction, you'll get a general recap of what the module is going to cover and maybe some new information regarding that module. Next is the required content. So required content is content that you are, of course, required to do. These include watching videos and doing the assigned reading assignments in order to take that quiz at the very end. For some of you, you may be very, very familiar with the material covered within these Canvas courses, so you can kind of skim through the videos and the reading if you need to, and immediately take the quiz. The quizzes do have a limited number of chances. I'll get into that in a bit. Just keep that in mind, though. Also within each module is optional content. These include video recordings and also reading assignments as well. This is really just for the overachievers or the people who are interested in the content itself and don't want to blitz their way through the course. But if you find the material interesting for that certain module, by all means, learn it, because it will come in handy later on the line when you're doing the mini project. There are 16 modules total. Each module, I estimate, would take around 30 minutes. It really just depended on how much knowledge you have. But I would say on the upper end, it should take around 30 minutes. The reading assignments within each module are predominantly through the lab manual. There may be a few here and there things that you're going to read outside of the lab manual, but for the most part, we want to follow what's in the lab manual so we can all use the same protocols and know what we're doing with each other. The videos are going to be a mixture of in-house recorded videos from a few years ago that the lab team recorded, and also public access. If you have a problem with these videos, if you cannot access them, if they're like 404ing or something like that, just contact the lab manager through the Discord or through email. They'll find a suitable video for you to watch or a suitable reading assignment for you to read. Also, the in-house recordings, I do believe, are slightly rough. So if you find that you're not getting enough information from them, just tell the lab manager. The lab manager can re-record those videos in order to bring them up to snuff. Finally, there are quizzes within each section. You must score 100% within these quizzes because we want to know that you understand the material that you're learning. They're not very hard, though. They're, like I said, they're a mixture of multiple choice and matching questions. You have 3 chances. If maxed, you can email the lab manager and the lab manager can email the advisor to get those reset for you. Just know I highly recommend if you miss it twice and you're on your 3rd try, just go ahead, email the lab manager through, message them through Discord and ask them, tell them what question, what quiz you're on so they can help you so we don't have to go through the whole process of getting your quiz reset. Like the goal is not to trip you up here. It's not to trick you. It's to get you learning the material. So asking questions is much more preferable than to have to reset the assignment in its entirety. Okay, I thought I'd include some screenshots for the module and the Canvas for Trainees. Over here is an outline of the different modules. So lesson 1's introduction. You have your introduction to the iGEM lab training program. Then it goes to the required content. There's going to be a bunch of links and videos within this. And then optional content, same thing. Finally, you have your quiz. It says 70 points, but don't assume it's 70 questions. 70 points just means that there's 7 questions worth each 10 points apiece. And you have to score at least 70%, so 100%. This follows by the lesson 2, which is the lab safety and behavior. We take lab safety very, very seriously within iGEM. We work with chemicals and microbes, and bringing those home to people who are immunocompromised could be a very big problem. So just keep that in mind, take your safety protocols seriously. And then it just keeps on going through the lab manual, pretty much the Canvas course does. It hits the labeling in the lab notebook, which you'll be keeping in throughout the course. And then finally, it's going to start hitting some fundamental like iGEM concepts. So like what's DNA? How do you cut DNA? How do you add DNA together and ligate it? All that sort of stuff. Here's an image of the required content. So you can see one is there's a document that's attached. Then there's a video that you can watch. And then it says read certain sections out of the lab training manual. The lab training manual is just our lab manual. So you'll find that on the Google Drive or also on the iGEM lab documents channel. You just go to those pages, read up on the required material, and then come back and take this quiz. And as you can see, this is when I took the quiz for the first time when I was going through this training process. Attempt 1 took me 2 minutes. I missed 1 question, then I retook it, and it took me 1 minute. So these things won't take you very long. Just go through them. I think when I took this, I actually finished the entire lab training course within a week. So that is, you can do that. I recommend splitting it up through some days so you don't get burnt out or anything like that. Okay, so this is really important. It's the lab manual. It's the main source of information that we have within the lab committee. The lab manual includes a variety of different things. These things include maps, lab notebook guidelines. Maps include like where stuff is in lab, what are the exits, stuff like that. Lab notebook guidelines include how you're supposed to write your lab notebook when you come to lab every day or every day that you come to lab. It includes format and the stuff that you should include in that. And then the lab equipment protocol and uses includes all of the equipment that we have available to us, which we'll be getting new equipment in the upcoming years, and also the protocol on how to use those. Of course, some of these protocols are slightly vague, so if you have any questions, we don't want you fumbling through the dark when you're messing with these. Ask the leaders, ask the independent lab workers, the lab manager, how to use these things so you don't mess something up. Then there's what I find the most important is the genetic engineering background info and also the various iGEM protocols. So you could skip straight to the protocols, but you wouldn't really understand them, which is why we also included the genetic engineering background info. Here you'll learn about the many different types of genetic engineering, the different types of cutting DNA, what we do in lab, how to transform bacteria, what bacteria that we work with. All that sort of stuff is given in the genetic engineering background info. And then once you read up on this, which is pretty much the point of the lab training course, you can finally start understanding and doing the protocols within lab. And so, which brings me to the Canvas course again. The Canvas course is pretty much just a sequential lab manual. This is where you're going to learn the ins and outs of how to look and find stuff in the lab manual, and also just learn the basic concepts in order to do these protocols. Here's the main thing. Learn basic biology knowledge, but you also want to know where to find everything else. So you need a protocol, but you don't remember it off the top of your head. Know where it is in the lab manual, know how to find it so you can find it when you're in lab in order to do it. Most of these techniques that I'm talking about, these protocols, you're going to be picking up in the mini project assignment, which is something that comes after the lab training Canvas course. Don't worry about that now, though. Worry about that once you start hitting the end of this Canvas course. The goal is to get you slowly eased into this, not to drop a bunch of stuff on you. Okay, so iGEM fundamentals. This is a page detailing what the general fundamentals look like within the lab Canvas or within the lab manual. These can be found starting in module 7 in the Canvas. This is when we start hitting on what's translation, RNA, DNA, and it's found in pages 30 through 38 in the lab manual. So this is like translation. It details what translation is, maybe a bit of information regarding what happens during translation. Same thing with RNA, and background information with regards to what DNA is and how to work with it. Which brings me to DNA. So within these videos, I'm going to be covering the fundamental concepts of what DNA is, and further on what proteins and how to work with them. In this first video, I'm going to be covering DNA, RNA, I think, translation, and transcription. So DNA, just for recap, is the code of life and is programmed through chemical compounds. These chemical compounds are adenine, which is symbolized A, guanine G, cytosine C, and thymine T. In RNA, these are different. Though I'll get into that later. One thing to know is DNA is double-stranded. So as you can see here, it's like a there's two backbones, right? So here's your nucleotides, and then it forms like a double helix structure, right? So that's the double-stranded nature of DNA. Of note, these nucleotides G, C, A, T, they bond to each other in this specific way. So adenine bonds to thymine, cytosine bonds to guanine, and these form base pairs. And so once you form a base pair, you can put these in any sort of sequence, and that can form sort of like data. So it's very, very similar to binary, which is why people relate like computer science to biology all the time, and why computer science majors do generally pretty good in biology, is because that you form a sort of code of life that your body that your cells can read in order to code for certain proteins, so your body functions a certain way. And this is how the base pairs form. So you can see that there's hydrogen bonding between these two base pairs, and that's how they stick together. I'm sure you went through hydrogen bonding in Gen Chem 1, but that's what keeps them together. And then there's much more, there's the phosphodiester backbone that holds these other ends together. Those are much more solid than this hydrogen bonding. Okay, continuation on DNA. So DNA reads in a specific sequence, and this is just like an arbitrary assignment that we've given to it. So we start at the 5 prime, which is the upstream strand, and then we go down to the downstream, which is the 3 prime. So you read it from left to right, with the 5 prime on the left and the 3 prime on the right. It's very similar to reading English in a certain way, and of note, every 3 nucleotides is a codon. Codons are like little segments of how we read them in the greater scheme of things. Codons code for proteins later on in the sequence, and we can generally split a DNA gene into different codons that code for it. So like this pair right here would be a codon. So now that this DNA is formed, and we have a way to read it, and we have this standardized data storage, we kind of need to figure out a better way for your body to read it. So we have this arbitrary way to read it, but how does your body actually get the enzymes required to go through this, read it, and then create the proteins that are required to make your body work? That brings me to RNA and transcription. So transcription happens within the nucleus of each of your cells. And this is where this enzyme called RNA polymerase, it's a protein that comes and it starts reading the DNA from the 5' to the 3' direction. So this RNA polymerase binds to the DNA. The DNA starts unzipping, so those hydrogen bonds that I mentioned before starts breaking. And as it goes through this, it starts assembling an RNA copy of the template strand. So what very, very important here is that the 5' to the 3' direction left to right is the coding strand, and the 3' to the 5' direction left to right is the template strand. So when this RNA polymerase binds to the DNA, it attaches really to the template strand, and where it starts attaching nucleotides, complementary base pairs of RNA to this template strand. And because the template strand is like a reverse copy of the coding strand, the reverse copy of the template strand is just a duplicate of the coding strand. So the best way for this to create this RNA copy is to get the template strand, so you get a one-to-one, not really a one-to-one, but a pseudo one-to-one copy of the coding strand. Why you say pseudo is because the T is replaced with uracil, which is just a different type of base pair. RNA is made out of that, so you replace the thymine with a uracil. And after you make this DNA strand, it cleaves it. The RNA strand. The RNA strand leaves this whole structure, and it leaves the nucleus. It goes through the pores within the nucleus, and it goes out to the endoplasmic reticulum where the ribosomes are held, and it starts binding to those ribosomes in order to create proteins later on the line. This RNA is typically called messenger RNA. There's many different types of RNA within our cells, but it's called messenger because it brings the DNA template all the way from the nucleus to the endoplasmic reticulum where you start creating those proteins. So next is translation, and this translation step occurs within the ribosomes, or I guess within the endoplasmic reticulum where the ribosomes are located. So it starts when the ribosome attaches to the 5' end of the mRNA that went to it. So as you can see, it attaches here. And then another type of RNA, which is called tRNA, matches with the codon. So this ribosome helps this tRNA find the correct codon, and it attaches to it. So as you can see here, the AUG on the mRNA corresponds to UAC, because that's the complementary base pair or the anticodon. And each of these tRNAs hold a amino acid at the end. And so once these come over, they start matching and attaching due to the ribosome. Once it matches, the ribosome cleaves the amino acid off of the tRNA, and the next one would come in match, and then this amino acid would attach to the next amino acid, forming a chain as it goes from the 5' to the 3' direction. And so this long chain of amino acids that's forming as these tRNA matches to the mRNAs, these long chains start forming proteins. And so basically just a protein is a long chain of amino acids that's formed in a specific 3-dimensional and 2-dimensional structure. A long chain of amino acids is called a polypeptide, and then these polypeptides combine together, and they change 3-dimensional structure, and those form proteins that do pretty much everything in our body. Okay, so in summary, this is called this whole sequence that I explained is called the central dogma of molecular biology. It starts where DNA is, and DNA is self-replicating. So your cells don't die off. They can create products, which is themselves, and so your body can continue on living. So they replicate, and then in order for your body to use that DNA that's stored in order to code for other chemicals, other processes, it has to do transcription, where the RNA polymerase comes in and it starts creating mRNA. That mRNA leaves the nucleus where it goes to the ribosomal units. These units start translating the mRNA into tRNA and amino acids. And these amino acids form together in order to form polypeptide chains, which therein form proteins in the larger structures. So that was the general recap of what DNA, RNA, and transcription translation is. I'm sure you learned most of that in your high school biology course. But next time we're going to be covering a little bit more in detailed stuff. So what enzymes are, what plasmids are within bacteria, and what the bacteria that we use in lab is. We predominantly use E. coli. I also will be going into the parts, the registry, and also maybe just briefly touch on the mini project in order to get you guys comfortable with how lab committee is structured. I included some pictures here of what we're going to be doing next time. This is a picture of the registry where we find most of our DNA for iGEM. This is the location of where it is in the kit plates that they send us. Primary protein structures. So these are the amino acid chains that form to form that protein. This is a plasmid that's found in many, many different types of bacteria. It's how they replicate their DNA strands and how they give their DNA to other bacterium. And this is just a pigment of bacteria, pretty much. This is growing on an agar dish. This is, I believe, is a picture that we took a little while back in our lab. So that's all I have this time. If you want to know a few of the projects that we covered in the past, I have included them through these links. During these presentations, there's going to be quite a few links. If you want to follow these links, just go to the description of whatever video you're watching. There should be a link to the PowerPoint presentation. Then you can click on those links and follow what I'm talking about. So these are just a few of the different projects that we did in the past. In 2019, we did Missouri S&T and the geneticist cell, which is more of a human relations project. Through 2020 project, due to COVID, we extended our deadline to every 2 years because it was an online competition. We didn't really want to take part of that. In 2018, we did the bee tree, which was creating a resistance to the emerald ash borers and ash trees. Contamiplant was a diagnostic tool where plants, you'd put them in water, they'd try to find any contaminants, and then they change color. And then our most successful project, I say, was the white nose syndrome project, where we were trying to create bacteria that helped bats fight against the white nose syndrome that's very much plaguing bats within the Missouri cave system. And that's all I have. Thank you very much. See you next time.